should stop sharing.
Okay, can you hear me?
Yes, we can.
Yes.
Oh, fantastic. Okay, so I will now share my screen. Okay, so thank you very much for the kind words of introduction. It's a huge pleasure and honor to be here today. I have to say, until a week ago, I did not imagine I would be giving any presentation anytime soon. I'm currently on maternity leave. So it's really great to be able to speak here. So I will take the opportunity to show you some of the work that I did in my DPhil. And the main focus, okay, now can't move here. Okay, so I'll have to do this manually, that's fine. Okay, so the main focus of the talk will be the application of persistent homology to tumor blood vessel networks. And then I'll give a tiny flavor of applications of local persistent homology in the final minutes of my talk. All of the work that I'm talking about is written up in manuscripts, which you can find online. Okay, so what is persistent homology? We are more likely to probably have come across the term topological data analysis over the last few years. It's grown quite a bit. And this is an umbrella term for algorithmic methods that are used to address questions about high dimensional data in a quantitative manner. And it's in particular a tool for understanding shapes and surfaces in data structures. So topology is known to be very useful. For example, if I give you these two cheeses and you're not allowed to comment on taste or smell, you can still tell me that they're different because one has holes and the other doesn't. And this is exactly what topological data analysis, sorry for jumping, can tell you about these two cheeses. Persistent homology is a mathematical formalism for analyzing topological invariants such as connectedness, loops, and holes in various dimensions. And it's one of the methods from topological data analysis. And we don't just learn something about the shape of data, but also how much certain aspects of the shape persist in this data set. We look at local information to get a global idea of the whole data set. And in particular, what is interesting in the context of networks is that this allows the analysis of more than just pairwise connections. So what is the shape of data? Well, if I were to give you these 15 data points and ask you what their shape is, well, your initial response will probably be, well, they're points, they don't have a shape. But then you might start squinting your eyes and tell me, oh, they're arranged in a loop. And this is exactly what you can see using persistent homology in this data set. So how do we go about this? We take the points and then we blow up little balls of radius epsilon around every data point. And as soon as two of those balls intersect, we connect them by an edge. So right now we can't see any loop. We increase the radius a bit, and now if we have three pairwise intersections, we will connect them by a triangle. If we have four, then we connect them by a tetrahedra and so on. So you really include higher order simplices as well. So right now you can still not see the loop, so let's increase the radius a bit. Okay, now we finally have this loop that I've been talking about. However, if we choose the radius to be too big, everything is covered up with triangles or higher order simplices, so you can't see the loop anymore. So how do we know what radius to choose here? Well, the key idea in persistent homology is really you don't have to choose one radius, but you choose a whole sequence of them, ideally all possible radii. And you study the changes of the topological invariants along this sequence. So the sequence of radii gives you a so-called filtration. This is a sequence of embedded simplicial complexes. And this along this filtration, you then study in dimension 0 connected components, you study in dimension 1 loops, and in dimension 2 voids. So what I showed you just now is a so-called Vittoris-Rips filtration. And this works a little more formally in the following way. You choose a sequence of increasing distances epsilon I, and in the ith filtration step, you define the k simplices to be the unordered k+1 tuples of pairwise distance at most epsilon I. Now, there's a very nice visualization which you can use to keep track of what is happening to the topological invariants along your filtration. And this is a so-called barcode. So on the x-axis here, we have filtration values. In the case of the torus Rips filtration, this is the radius epsilon, which you use. And then every bar represents one topological feature. So this means in dimension 0, every bar represents one connected component. And initially, you can see that you have 15 separate bars. And then as we increase the radii, some of the bars start to die because some points start to be connected to other points. So the connected component associated merges, and so one of them dies and the other gets to live on. And this happens until we have one connected component when all the points are connected in one single component, which then lives until the end of the filtration. In dimension 1, what we are looking at is loops. And so this bar represents a loop. And it starts when the loop first appears in the filtration, so at this radius, and then ends when the loop is covered up by higher order simplices. Now, the pipeline for persistent homology is you take some kind of data. This can be point cloud data, as I showed right now, or as I will show in a second, tumor blood vessel data, or I also looked at functional networks created from fMRI time series in other parts of my D field, or even the output of mathematical models. So you can really use a variety of different data and of different data types here. You feed these into the filtration, and then you get your barcodes, which you can analyze, again, with a variety of different methods, which I'm not going to go into detail here. But essentially, you can vectorize the outputs and also feed them into the standard machine learning tools or classification tools for analysis. So why is it that I'm interested in tumor blood vessels in particular? So a tumor, when it grows, at some point, it needs its own blood vessel supply. And it does that in a very clever way. It uses the vessels that are already around it in the body and signals to those vessels to grow new capillaries into the tumor, forming the tumor's blood vessel network. This process is called angiogenesis, and it is one of the hallmarks of cancer. And it's in particular a necessary step for invasion into other tissue and the growth of the tumor beyond a certain size. So it's a very important step and enables the tumor to become malignant. Functionally, these vessels have important functions to fulfill for the tumor. So they supply it with nutrients, but they also lead to physical support for the tumor. And they're involved in many more things, such as, for example, immune suppression around the tumor. Structurally, the vessels that come from this process, they look very abnormal. So you can see pictures here. And because they basically come into existence because of changes in the equilibria of pro and anti-angiogenic factors, which are quite chaotic changes, they look very abnormal, like they're large and variant in diameter, they're irregular in their branching. There are even shunts between arteries and veins, loops in these networks. In terms of function, the blood flow is heterogeneous, so you can have blood flow in different directions in the same vessel at different time points. And in particular, what you can see here is that the vessels are highly tortuous, which again is not very efficient. So what I wanted to do here is to use persistent homology to quantify the shape of these vessels, or even the tumor, and in particular also the degree of abnormality in these networks. And I measured this degree by looking at the tortuosity and the loops. And then also I want to get an idea how heterogeneous the networks are. So the challenge here was really to find a filtration, because there are not many filtrations out there. There's not a library where you can look them up. You really, for most problems, you have to come up with them yourself if you're beyond sort of standard problems. And so the challenge was to find a meaningful filtration that would be not only mathematically correct, but also helpful for biologists. So what I did was I came up with the radial filtration, and this works in the following way. So you define a tumor center. In this case, I take this to be the center of mass of points sampled from the tumor blood vessel networks. And then I kind of blow up a little sphere around this tumor center slowly, stepwise. And whatever is in this radius, so all points and edges, if both of their boundary points are within the radius, are included in the filtration. And by stepwise growing the sphere, I include more and more of the network in the filtration and slowly build this up stepwise. What this gives me is information about tortuosity in dimension 0, because if you have very wiggly vessels, then you can imagine them intersecting with the sphere multiple times, creating short connected components which quickly merge with the rest of the connected components. So this shows up as short bars in the corresponding barcodes. In dimension 1, what we have is loops. And because we're looking at all of this with respect to the tumor center, we also get a spatial resolution of the loops in the tumor. So the pipeline that we have here is we have data, we feed this into the filtration. So I showed you the radial filtration. I also use the second filtration to look at dimension 2 topological invariants, but I'm not going to go into detail here. I then create the barcodes from these filtrations. And from these barcodes, I extract topological descriptors. So from dimension 0, as I mentioned, we get tortuosity via the short bars in the barcodes. Then from dimension 1, we get the number and distribution of loops in the tumor. And from dimension 2, from its other filtration, we get the size of voids. So these are avascular regions in the tumor where there's no vasculature. I have two very different data sets here. So what they have in common is that they come from mouse experiments, and these mice were implanted a tumor. And then we have data from Oxford, from the Oxford Radiation Oncology Group, which is imaged using intravital microscopy. And this means that the mouse is alive while the imaging takes place. So these mice have a little window on their back just above the tumor. And the tumor is then imaged through this window. So, and you can even see the same tumor and its blood vessels over several days. So you can see how these blood vessel networks develop over several days. So this is really quite unique. The downside is you only kind of have the surface of the tumor because you're imaging on top of the tumor. Then the second data set comes from my pharmaceutical collaborators at Roche, and this is an ultramicroscopy data set. Here, the tumors are extracted from the mouse and then imaged via optical sectioning, and you can then do a 3D reconstruction. And the nice thing is you really get a picture of the entire network in the tumor. Now, both data sets have some control mice and then some mice that have been treated. So in both cases, we have vascular targeting agents that have been used to treat the mice. This means that these are drugs that directly influence the vessel growth and vascular architecture. In the Oxford dataset, we have DC-101, which reduces vessel sprouting and makes the blood vessels a little more normal looking. And then anti-DLL4, which increases vessel sprouting, so makes the whole thing look more abnormal. In the Roche dataset, we have bevacizumab, which is a common cancer drug, and this also acts to make the blood vessels more normal in the tumor. So for these treatments, we know how they influence the vascular architecture. And in addition, in the Oxford dataset, we also have treatment with radiotherapy. So we have one group that has been treated with a single dose of radiotherapy, and another group which has been treated with 5 small doses, so fractionated dose radiotherapy. So from the radiotherapy, we don't know how this affects the vasculature. So it's very interesting to see the results here. And just to stress again that the data sets are really quite different. On the left-hand side, you've got the Oxford data set. So you can see that there's a small network there and it's fairly shallow, whereas on the right-hand side, we have the Roche data set, which is a bit less high resolve, but then you've got the entire tumor there and the huge network to work with. So let's see some results. Here we're looking at the Oxford dataset, and we're looking at the treatments that have known effects on the vasculature. We're considering now tortuosity and loops in these networks, and we've got controls here in beige, and then in red we have DC-101, which makes the vasculature more normal looking. and anti-DLL4, which makes the, in pink, which makes the vasculature more abnormal looking. And you can see that for tortuosity, for example, here we've got the days since treatment. So on day 2, you can see that the pink group really has increased tortuosity versus the red group, which has been treated to be more normal looking vessels, which has reduced tortuosity. And for loops, you can see an even more extreme effect. So here in pink, for the more abnormal vascular structures, you can really see how the number of loops goes off and becomes quite high. And the controls also increase the number of loops, but not quite as much, especially on day 3 after treatment, whereas DC101 really stabilizes the number of loops over time. Here we've got the spatial resolution of the number of loops. So we have the tumor center and then spherical shells around it, and then different treatment days. And you can see how in the different spherical shells around the tumor center, you will have changes in the number of loops happening over those days. And you can see that, for example, DC101 acts fairly on the whole of those spherical shells, so on the entire vascular network of the tumor, with stabilizing or reducing the number of loops. Whereas DLL4, for example, on day 3 and 4, you can really see a change on the outside, mainly of the vessel network. For the Roche data, here we consider descriptors of loops and voids. And we've got the controls in beige, and then here in light purple, the treated tumors. And again, we've got the days since treatment. And you can see that the number of loops really goes down after treatment, and then after some time, it goes up again a little. And for the size of voids, so these are regions without vasculature, you can see that, again, as one would expect for the treated cases, these become bigger over time, whereas the controls sort of have a stable size of avascular regions. Now, for the unknown effects of radiotherapy, we're looking here at tortuosity and loops, and we've got fractionated irradiation in brown and single dose irradiation in blue. And you can see that for, in particular, fractionated dose radiation, you can see an effect that is kind of more long-lived than for single dose irradiation, where tortuosity even increases after some time post-treatment. In the number of loops, it's quite interesting. So the fractionated dose radiation, again, kind of number of loops goes up a little and then goes down again over time and sort of is stable, whereas for single dose irradiation, you have a variety of different reactions across different tumors. So this was very interesting to see. And again, spatially resolved, you can see that fractionated dose irradiation really stabilizes underneath the outer shell of the tumor, and then single dose irradiation kind of leads to increased number of loops, well, almost everywhere apart from in the core. And just to stress that these really are different parameters than what biologists have been looking at traditionally. So here you've got in black the traditional parameters that are being pulled out, and then here in orange, the new ones. So the loops that I showed you were loops divided by number of vessel segments. And you can really see that they form, in terms of correlation, a separate group to existing parameters that are being analyzed. Okay, now in the final 2 minutes, just a very brief outlook on applications of local persistent homology. So some of the computations that I showed you, or actually a lot of them, took 2 years to compute on the very best computers in the math department. And this is because computationally, persistent homology is still very difficult to do in many cases, especially for large and noisy data sets. And so I started to look into pre-processing or local applications of persistent homology. And it turns out that if you have a data set, for example, where you have points that are sampled from a sphere, these you would consider to be signal points, and then noise points around them, just by looking at local neighbourhoods of the points, and again, computing persistent homology on these and looking at their barcodes, you can extract a score that is differently distributed for different types of points in this data set. So you can see here that the distribution for sphere points and then noise points is quite different. We also have points that are generally very far away from other points. So these kind of form their own group. And based on these scores, you can then start selecting landmarks that are representative for the data set, and on which you could then run persistent homology algorithms to get an idea of the shape of the whole data set. And using very similar ideas, it turns out you can also, just by looking at local persistent homology around points, you can detect points that are close to intersections when they are sampled from intersecting manifolds. So here we have the conformation space of cyclooctane, which turns out to be the union of a Klein bottle and a sphere intersecting in two rings in R24. Here you've got the projection down to 3D. And we can really find these intersections or points close to the intersections using local persistent homology. So I hope that I've convinced you that persistent homology is very useful. It allows for the tumor blood vessel network as a first step towards relating form and function. The topological descriptors validate and quantify known treatment effects and show effects of radiation treatments on tumor vasculature, including also spatial resolution of such effects. And local persistent homology is very useful for landmark selection and can robustly also detect points close to geometric anomalies. And I'm sure that there are many more future applications out there. And I would just like to thank my supervisors and collaborators, which have been with me on this research journey. So this would be in particular Heather Harrington, Jared Tanner, Helen Byrne, Vidit Nanda, and Mason Porter, and then from the Roche side, Florian Lipsmeyer, Franziska Mech, Sato Nakuri, and then all the biological collaborators from Oxford Radiation Oncology, and finally all my funding bodies who funded my PhD. And thank you very much for listening.
Thank you.